Breautium. Okay, so I think we can start. Well, 1 minute. This is for the questions. So maybe we can discuss that after, but I know it's very annoying to come with a strike. I live here 3 years. You don't need to explain me how annoying Sao Paulo is when these things happen. But there is a but, and the but is that, yeah, well, we have the course, it's 1 week, and tomorrow, well, we'll have to go through it, and once we are done, we can go to the bar if you want, relax with a beer, or go back to the hotel, or do whatever. But sometimes life comes like this, and I don't think at this time I have to explain that. There are things that we cannot change, and I guess many of us here have got financial support to be in Sao Paulo, no? Otherwise, I would be teaching from my apartment and not here, no? So I would ask you to come. There is projects. The projects are very important. And yeah, please, let's try to stop the conversation there and just try to make it as easy as we can among all of us. Great. That being said, let's start. So I changed a little bit the plan that I had. I'm going to cover the same things. I don't know if I'm able to do something else, so I'm going to cover the same things. What I want to do is...
que legal! Botou a gente cantando. A gente me inscreveu só as nascidas em 2001.
A gente pode continuar, André, que tá todo. Só deixar aqui.
Ai, que legal! Eu assisti.
Não, mas eu não vou assistir a aula, não.
You see, that's why I don't like teaching with Zoom. Yeah, anyway, I think I'm very old. So what I want to do is I want to discuss with you the importance of considering space when we study an ecological system. But the points I'm going to make are very general. So most of the work I do is regarding the importance of space in living systems. I said before, we study animal movement, we study spatial distribution of vegetations, we study microbial community ecology. And I really like understanding what is changing in these systems when we consider that all these entities that are interacting are somehow distributed in a space, instead of being all interacting with all of them. So I want to start showing you some examples of why space is so important and why we are so interested with space. The first is what you're going to see here in the screen is the trajectory of wolves in the US. So this is something that is very common now in ecological research. People can put GPS on animals, usually in the neck. They put collars, and they are continuously emitting the position, no? And then we know where they are, and we can study how they move. It's more or less the same things that Samsung, Apple do with us when they put a phone in the pocket. The only difference is that we use the data for something more positive, let's say, than these companies. So you see these are trajectories, so different animals, they are moving, they don't move all over the place, no? So they have their places where they like to be, as us, no? I don't live all over the place. There are some regions where I like to be, and that of course is going to change many things, no? Of how these wolves are going to behave. It's very clear that this wolf here will be very unlikely to find this wolf there if they are not in the same place, no? So how many of you have ever studied anything related to population dynamics, let's say? How many of you know what is the logistic equation? Okay, we have a good starting here. So the most common equation to study these kind of ecological systems is what people call the logistic equation. This is nothing else but a quadratic equation in which we have some linear term that is going to be the most important one when the population is very small. And then we have a quadratic term that is going to avoid this population to grow indefinitely large. How many of you find this equation problematic in any way? Like, how many of you would say, I don't like this equation? It's problematic? I mean, I can solve it analytically. We can solve this equation analytically. So in principle, that's not a problem. Mathematically, it shouldn't be a problem. The problem is that most of these models here what they assume when they, I can rewrite this equation in this way, no? So basically, all these models, when they introduce these kind of nonlinear terms here to account for interactions, they are doing that based on a very, very strong assumption. It's that the population is well mixed. This is the law of mass actions that maybe many of you studied in chemistry, right? When we have reactions. So basically, what this equation is telling me is that if these things here are metro drivers on a strike, the probability that they find each other is equally likely with any other person in the room. So the probability that my wolves are going to interact with any other wolf is the same. It doesn't matter who the wolf is. And of course, when we have data like the one I was showing before, that becomes problematic. And it's even more problematic because this is not the rule. The fact that these animals are distributed in space is not an exception. It is the rule. So here we have same kind of trajectories from a very cute white-faced monkey in Panama. And you see that they also have their territories and they don't overlap perfectly fine. And this is the norm. When you have animal tracking data, this is the norm. Of course, if you think of plants, well, good luck thinking that plants are well mixed, no? So I am not saying that these models are wrong. Well, I'm saying that these models are wrong. They are wrong, but I'm not saying that they are not useful. What I'm saying is that we need to understand when they are useful and when they are not, no? So what is the limit at which we can stop, or we must stop using them? And that is something that I am very interested in doing. So the way I thought I was going to organize the course is by trying to tell you a story. I like courses when they tell a story, and then we can take parts of the story to learn. A story in which you will be able to see in which limits these kind of equations are good, they are okay, they are fine, and in which limit they are not. And in building this idea, we are going to learn what are the tools that we need to study these models and the new models that we are going to evaluate, okay? And if you have questions, please stop me at any time. So this is how I am going to organize the course. We have 4 classes. In the first 2 classes, I'm going to give you an idea of this. I'm going to introduce you the biological system that is going to tell us the story. I'm not going to tell you the story. This system is going to tell us the story. And in the last 2 classes, I am going to use a much simpler model from statistical physics, probably the simplest model from non-equilibrium statistical physics, that we are going to use to learn the tools that we would need to analyze this system when we go to get closer to reality. The reason why I'm doing that is because this system is very, very complicated. So I'm going to give you the papers in which they do what we are going to do for the simple case in the realistic case. But you will need to sit down and understand the equations because it's like studying a system in 1 dimension or in 2 dimensions. So we are going to do the simple case here. And my goal is that with the things that you are going to learn in these 2 classes here, then you will be able to go to these papers if you want and understand them, maybe studying them for 2 weeks instead of 2 months. That is my goal. I needed 2 months to study those. So the papers that I think will give you a very good idea of how the course is going to be are these 5 papers here. So this one from 1981 is the experimental one that we are going to discuss, the number 4. Number 2 is a general idea of how we can use this kind of lattice models that we are focusing on the course in ecology and also in social sciences. I know now there is a lot of people from quantitative background interested in social systems. And then number 1 and number 3, no, 1 and 5 are theoretical attempts to model this experimental paper. And number 3 is like a review on mathematical techniques to study these models, okay? If you are interested in lattice models, probably from a physics point of view, this book by Marro and Dickman is, I wouldn't say the Bible, but a very important reference. And this other paper is less physics and more applied mathematics. More or less the same techniques, but in this book, number 2, you are not going to be finding things like, I don't know, critical exponents, universality classes, and a lot of statistical physics jargon that you will for sure find in number 1. So it's the same concepts, but with different approaches. If you like physics and statistical physics, then number 1. If you are trained in something else, then number 2 maybe has the information that you want. So before starting, how many of you are physicists, mathematicians, or something similar? How many of you are not? Okay, perfect. So I'm going to do math in the Blackboard, but I'm not going to be extremely rigorous with my math. I want to help you building intuition with the math. So if you get lost at some point, you can ask, and I can go back. And if you get lost with some calculation, I will always point out to you which is the important part of the calculation, which is the intuition that I want to build with this math, okay? I'm not going to throw equations in the Blackboard without, or at least I'm trying not to do that. If I do that, let me know. Great. So let's So let's introduce this story that I was mentioning before, this experimental paper from the 80s. So we are going to be thinking a little bit about bacteria. Bacteria are very, very tiny organisms. They are single cells. And for many years, people thought that all the things that they were doing were dividing, growing, and dying, and they were very boring organisms. Now we know that's not the truth. They do very interesting things. And we are going to see one of the things that they do, and we are going to try to understand how they do the things that they do and why we observe the things that they do. So we are going to study a system in which we are going to have bacteria of 2 types. For those of you that are already familiar with biology, I'm going to tell you that this is E. coli. The bacteria that we are going to work with is a very common bacteria to do experiments. For those of you that are farther away from biology, this is just a bacteria. And they have 2 different phenotypes. There is 2 different types of this bacteria. What we are going to call producer bacteria, they are able to release a chemical out there that is able to kill the other type of bacteria that we are going to call non-producers. So basically we have 2 types of bacteria. One is able to release toxins that kills the other bacteria, okay? So people were very interested in understanding this process from a biological point of view. Now I'm only introducing the biology. Because when these bacteria, they secrete these toxins, they can also be bad for themselves. So you can think that these bacteria, whenever they are releasing these toxins, they are somehow opening doors in their membrane, outside here. They are opening doors, and they are releasing chemicals. Because they open these doors, these chemicals can also enter the cell and can kill them. So this kind of toxin-producing behavior, it's on the one hand, is good because it allows them to kill competitors, to kill other bacteria and allow them to grow more. But it's also bad because it can kill themselves. And this is something that in evolutionary biology, for people interested in biology, is a very interesting behavior because it's really hard to understand why we would observe in nature, why would evolution favor behaviors that are harmful for the individual that is executing it. No? So for instance, for those of you that maybe are interested in evolutionary questions, there is all these questions of the evolution of cooperation. No? So why people help each other, why organisms help each other, why there are some birds that they invest in growing the chicks of other birds instead of themselves, and these kind of questions. They are very important in evolutionary theory, and therefore this kind of microbial system was very interesting for biologists to try to manipulate it in the lab and find why this behavior was observed in nature. So what people did when they were doing these experiments, and let me try to follow the notes because if not, this is going to be a mess. They did a type of experiment that people do very often in microbiology. They grew these bacteria in a flash with some liquid with sugar for the bacteria so they could grow. And they put together some producers and some non-producers. They were all growing here together. Once they were growing a lot, they took some part of this medium here, and they did another transfer to a different flask, and they repeated this growth process many times. And at the end, what they knew was how many producer cells I had in the beginning and how many producer cells I have at the end. They were able to count the cells. Okay? So I can repeat my experiment saying I put 10% of producers, 20, 90, and then after how many days I try to see how many I have. So this is what they found when they did this experiment. What they found is if you look at time in the x-axis and proportion of producer cells in the y-axis, and maybe if I use this I look a bit smarter, they found that if you start this population with very few producer cells, they die. They go to 0. But there is some critical threshold here, and if you put more cells than this critical threshold, then they grow, and they eliminate the non-producer cells. This is so basically what in mathematical jargon, how many of you have taken a dynamical systems class? Okay, good. So this is a bistable system. I do have a region of initial conditions in which my system will go to a stationary state, and I have a different regime in which my system will go to an alternative stable state. Now I'm going to repeat my experiment in a different setup. Which setup do you think I'm going to use if we are in a Lattice course? Okay. So what I'm going to do is I'm going to take a plate, and instead of giving the sugar in water, I'm putting the sugar in the plate. And then I'm going to put my cells there, and they are going to grow. And they are going to do the experiment. Sorry about that. The experiment is pretty much the same. I'm going to start with a mix of cells, with producer and non-producer cells. I'm going to put a drop. The drop is going to grow. And once my drop is grown, I'm going to take a sample of this drop, and I'm going to repeat my experiment many, many times. And I'm going to see the proportion of cells before and after they grow. The only difference that I'm going to keep is how these cells are growing, how they are interacting with each other. How do you think these cells will interact here with each other? If they are all in a flask, and I am mixing that flask, they are well mixed. No? The probability that my toxins, if I release a toxin here and we are here like all shake, then the probability that they will arrive to the back is very high. But if I am here and I'm just dropping them, the probability that people in the back will be affected by my toxins is very, very low, right? That is the only difference. In this case, my system is well mixed, and in this case, it is spatially structured. And these are the results that we get for this experimental setup. Now, regardless of the initial frequency of producers that I have in my system, they always win. They are always able to, even though they are producing this toxin that kills them, even though they are investing some energy, no, in producing this toxin. Producing the toxin is, I mean, you need to put some energy. Even in that case, they are still able to always grow and eliminate the non-producer cells. Yes?
Those no-producing bacteria know that the chemicals kill them, or they have no information?
So they don't have a defensive system to react to the chemical, if that is the question. It is not like I feel it and then I don't react.
And they know that if they go towards the chemical, they die, or they have no.
So they don't do any chemotaxis on this chemical, if that is the question. They don't follow gradients of the chemical. Yeah. They are identical. These two cells are identical. The only difference is that some of these, if they are not releasing the chemical, they don't die. So imagine that this cell I am here, I'm one of these bacteria. And I open my doors to release the toxin. Once I close my doors and I stop releasing the toxin doesn't enter into me. But the other cells always have their doors open and always get the toxin inside. Okay, so the difference here is that these producer cells, for a little bit, they are sensitive to the toxin, and the others are always sensitive to the toxin. Is that clear? Because if this is not clear, then we don't have business here for the rest of the week. Okay. So any intuition on why we have this difference here? Any idea? No idea? They are localized, the toxin is localized, and therefore it's avoiding the chemical to go through the whole population and harm the other individual. But something that I think is very interesting of this result and took me thinking for a while is that in principle, if I am growing next to my neighbors, my toxin is killing me, no? It's killing my neighbors. So it's very hard to understand why this is happening, no? So to try to understand that, well, we're gonna do a model to try to understand that. But let me show you a little bit more of experimental figures. I think this is these figures are very pretty always. So what you have here is a picture that these people took from the experiments. This dot here is a colony of these producer cells, and what you have clean all next to it is a region in which, because of the presence of this chemical, there are no bacteria. They kill them. So even though this chemical is somehow for a little bit affecting these cells, it's also creating a space for the colony to grow, no? So I am not aiming that you are understanding what's going on here at this moment. All I want you to take at this point is that because of the localized growth process that I have here, I am changing the dynamics as compared to this case here. And we are going to do a model to understand that, okay? We are going to try to formalize these ideas and this intuition. So this is a clear example of why we need models, because with this intuition we get ideas, yeah, well, it makes sense, but then there is some inconsistencies in the arguments, no? Like, if the toxin is killing my neighbors, then it's killing my sibling cells, and this is not very, very clear. That's why we're going to do a model. Okay, questions? Yes?
So if you have not a circle inside of it, but you have it like randomly distributed somehow, so you would suspect the well-mixed behavior, right?
If you
change this condition, this initial condition, to a fraction of bacteria which are, let's say, spread over the place, so then you would expect that is well-mixed, right? And then the behavior would be the same as...
it's well mixed in the beginning, no?
Yeah, but that's my question.
Well.
So what would you expect? Right?
In the beginning is well mixed. I would expect that maybe I will favor the well mixed case, but we will see in a little bit with the model what happens. But that is a very good question.
So the geometry matters, right?
The geometry matters. In most cases it does. In most cases it does, because you are, inside this drop that I am putting, everything is well mixed. Right? So I am shaking my bacteria and I'm dropping a drop there, and they are interacting within the drop and they are growing. And as they are growing, they are changing interactions. So basically your question is, if I am forcing a well mix from the beginning, what is the outcome? Is it going to be the one forced by initial condition, or is it going to be the one forced by the dynamics? And this is a very fair question. I think it's a very interesting question. And that's one of the things that we use the model for, no? To try to understand how things would change. Great, so let's try to do a model for this.
And the toxin has like a decay time, or does it stay there forever?
No, it decays. It decays with time. That's actually depending on the characteristic time scale of the toxin. If it stays for a long time and it's able to diffuse further, then you are closer to the well mixed limit, no? Depending on how they interact with the space. Well, Now you have a quiz if you want. Wife. So far we have a research question. The question that we have is, well, we have an observation more than a question. And this observation is that somehow space is favoring these producers. And this is it's a very interesting observation. If you think about it, do you want me to write bigger? No? Okay. If you think about it in terms of the consequences of these two behaviors here, what I'm saying is that if I have a population of non-producer cells and I put a little bit of producers, so let's say I have a mutation that allows these cells to kill their neighbors, if they are well mixed, they have a problem. They are not able to percolate in the system. So if you have a stronger intuition instead of ecology, biology, you have a better intuition thinking about society. If you have a population here, and I drop a little bit of a fake news, a rumor, what this result is telling me, this biostability is telling me, is that if that is a very, very small frequency, it's going to decay. It's not going to percolate in the system, right? Here's the same. If producers are initially in a very small proportion, they die. But in this case, if they are in a very, very small proportion, they percolate. So it is not only that in one case we have biostability, in the other case we have more stability, and the phase diagram is going to be much more fun. And it's also that fundamentally, the ideas, the processes happening here are fundamentally different, just because of the space. Okay? Great. So let's see where I am. So let's try to build our first model for this system. Of course, I'm going to start with a lattice. And what I'm going to assume is that if this is my experimental plate, each of these cells, they can be in, let's say, 3 different states. One state is going to be occupied by a producer. State 2 is going to be occupied by a non-producer. And state 0 is going to be empty. And now inside my lattice, I'm going to have some dynamical rules. Let's say that in my initial drop, this is what I have, for example. And now all I have is that these cells, let's say the producer cells that are going to be the white ones and the non-producer ones are going to be the yellow ones. What these cells do? What are these cells going to do in my system? They don't move, sorry, they don't move. Let's forget about movement. They reproduce. They reproduce. Let's say that these cells have a reproduction rate that I'm going to call beta 1. They die. Let's say delta 1. And the same for these cells here. Beta 2 and delta 2. They only die and reproduce. I said before that these producer cells, they need to invest part of their energy into killing the others. No? They produce this toxin. So basically, in my model, this beta 1 has to be smaller than beta 2 because the non-producer cells, they are using all their energy to reproduce, but the other guys, they are investing a little bit into secreting the toxins and the rest into reproducing. So this is what usually in these models we call the cost. It's the cost of the behavior that we are going to study. Clear? And then for the death, I'm going to assume that they have the same death rate. What else do I need? Sorry? Ah, great question. How is my model killing here? Interaction. No, I need to do the killing. I need to do the killing, and my killing is going to be. I lost practice with organizing the blackboard. So let's say here I'm gonna say beta 1 smaller than beta 2 and delta 1 equal to delta 2. And I need the killing process. That's a great point. So basically what I'm going to assume is that whenever I have a producer that is somehow sitting next to a non-producer, the non-producer can die with a rate gamma. And this is the first point where I have space being very important, no? This guy has to be sitting next to the other to be able to kill it. Otherwise, I don't trigger this process.
So, but it becomes empty or?
It becomes empty, yes. Sorry, empty. Good point. I like when you are paying attention.
It is similar to automata.
Yes, what I'm building, great question. What I'm building is a kind of very, very simple cellular automata. Cellular automata, the last day, if I have time, I will show you an actual cellular automata we built in our research, so you see how things can get more complicated. This is a very simple cellular automata. That is the next point I was going to make, yeah. So I have rules. I have probabilistic rules. What is the first thing that I can do with these rules? Simulation. Everyone likes simulations. Ooh. I don't know if the building likes simulations. Everyone likes simulations, right? I can put these rules. I can start my lattice. And I can do, we will do it tomorrow afternoon, a kind of Monte Carlo simulation for those of you that are familiar. And I can start updating my lattice, and I can see how blue and white start changing and all these kind of things. That is the first thing I can do, yes. Who can tell me a problem of doing a simulation? Come on, someone that is a theoretical physicist has to hate simulations in this room. No? It takes a lot, it can take a lot. There is a very big problem, now speaking more seriously. The problem with simulations is that it's really hard to get general knowledge of the system. Because we throw some parameters, and we see what is coming out, but we don't know how general that is, no? And we don't know if our code is correct. No, it's serious. I mean, you would be surprised of how many papers are out there with bugs in the code, rules that are not very clearly explained, and then everything changed. So it's serious. It's good to have theory supporting the simulations because of those things. So of course, simulations are very interesting. They give us intuition. Sometimes it's the only thing we can do. That's why we need to learn how to do them properly. But they are even more important and more interesting if they have theoretical support behind them. So basically what we need is theory. We need equations that support the simulations and that give us an approximated intuition of how this system is going to behave. And this is all I'm going to be discussing with you during the week. So we are not going to cover a lot of material. I usually don't like covering a lot of material because I was not a very good student when things were very big, like I used to get to lose focus. I'm going to focus on very, very specific things. I'm going to focus on how we can approximate these spatial dynamics by systems of differential equations, and how can we analyze those differential equations. Okay? So I prepared material for what I thought was a week of lectures. If at some point there is some parts in which you think it would be better to stop and spend more time, I'm very happy to do that. Okay, I'm not obsessed with covering what I prepared. I prefer that you learn 2 things well than 3 bad. Okay, but at least you need to learn 1, because if not, this is a disaster. Great. So I have my microscopic rules, my stochastic rules in the lattice, and I want to write an equation to describe the state of this system. I want to describe some sort of macro state of the system, starting from what would be a microstate that would be given by which cells are white, which cells are yellow. So I have 3 variables here to describe my system. Now, the first one would be the density of P. I would have the density of non-producers, and I will have the density of empty places. These are global densities, okay? This is just if I look at my lattice, what is the fraction of nodes that is in each of these states. So in principle, these 3 variables are not independent. If these are fraction of sites, then basically the sum of the 3 of them has to be equal to 1. We all agree, right? So in principle, I can describe my system by 2 of them, and then I know that they add up to 1. Great. So let's write an equation for one of these. Which one do we start with? Well, we're going to forget this. This empty is boring. Which one do you want to start, 1 or 2? Okay, 1. Let's follow the order of my notes. So I am going to write an equation for how the density of places occupied by species 1, by producers, change. So the first thing that the first process that is contributing to the change in the density of individuals 1 is death, no? They die at a constant rate delta, and remember here, they are equal to delta, okay? And then I have a second process. This is death. And I have a second process that is birth. So the birth basically is going to be the rate at which these producer cells reproduce, that is beta 1, times the number of cells of the type 1, times what? What is, let's forget about the math. What is physically different, fundamentally different between the death of my cells and the reproduction of my cells? Space. That was an easy one. If we're in a space course, what is fundamentally different? There is a very important difference here. Death is just that I take this and I say bye. Reproduction is different because I can take this cell that is supposed to reproduce, but she cannot because the environment is crowded, right? So the birth, has spatial correlations here. So basically, the rate at which the reproduction of my bacteria is going to contribute to the change in the density is going to be the reproduction rate times the number of cells times the probability that I take one place and then I take an empty neighbor. This is a conditional probability. If I take this cell here to reproduce, and this probability is 0, because the environment is crowded, then this process cannot contribute. And this is a conditional probability. This is the probability that I take a 1, I take one of these, and then I take an empty neighbor. So here I have spatial correlations. So if this is birth, this term here is giving me the spatial correlations in the system. Is giving me the spatial correlations? No, sorry. Is limiting the growth due to the spatial correlations in the system. And this term is extremely hard to handle. It's very hard to understand, to get an idea of what is this probability. Why? Because it's a local property. So here I am mixing things. I am describing my system at a global scale by the total density of ones, but in that equation I am introducing a local property. Those of you that work in complex networks, for instance, how many of you work in complex networks? I guess a lot. Ah, wow, physics is changing, not so many. Good. Those of you that work in complex networks, this probability is going to be very hard because it's not even that the probabilities are local, it's also that the number of neighbors of each position is different. And that term can be a mess. Okay? Of course, I'm going to work in one-dimensional lattice because I am smart enough to avoid going into these messes. But the techniques that I'm going to show, there are papers that extend them to complex networks and that they do things like in more complex geometries and topologies. So if you want to know about them, I can send you references. Great. So we have equation for the density of producers, and now we need a place to write the equation for the non-producers. That we are going to write below. So let's try to make some room here. So this is death. This is birth. And now I'm going to write the change. Can you see this from behind? Yes? Okay. So what is the first process that, the first one I'm going to write it because I don't like cheating. That's what I used to answer in class always. Ah, that one. What else? Birth. The birth is not very different, no? So now, of course, I need to take a 2 and then an empty neighbor, no? What else? The killing. Sorry, there was something, someone saying something different? Yeah, of course. Yes, this is different to this. It's the probability that I take a 2 and then an empty neighbor. It's completely different. And then I need the killing, no? That I've been avoiding the whole class. Let me make sure that I use the same notation. If you want, then at the end or at some point, you can take photocopies of this. That's why I try to keep the same notation. So I need the killing. Who wants to try the killing? Anyone?
It will depend on
Row
row 1, right?
1 and row 2. Who said row 2? That's very interesting intuition. Row 2? Row 2? Raise your hand if you think I need a row 2 here. Ah, you all come from dynamical systems. I can only kill my neighbors. The probability that I take a 1 and the neighbor is a 2. Great point. We're going to do that in 30 seconds. I promise we didn't discuss the class before. So this is, for those of you that have worked with models in population dynamics, you already see the structure of this model. And if you haven't, it's not a problem. I mean, the structure is very clear. The only thing is that whenever I have interactions, my interactions now are local. So they depend on conditional probabilities between neighbors in my system. We're going to do that in a second. We're going to do that in a second. So this is my model. And in principle, this is the equation. These are the equations that I have to analyze. What is the problem related to what Mateus was saying? The problem is that I have here 3 conditional probabilities, that they are local properties of the lattice, and this is really hard to know how to handle. So basically, all we are going to do in the next, in the next couple of days is to try to understand how I can analyze those terms to get closed systems that I can analyze and I can understand. Okay, that is my goal. My goal is that you learn how to handle those terms and that you learn how to do these simulations. The simulations, usually we try to focus a lot the courses into the math and getting the equations. I want to use the afternoons to also for you to learn how to handle these simulations a little bit. Those of you that already know can help the others and so on. Great. So let me see. Perfect. So before we try to approximate those terms to get an idea, we said before that the global densities are not independent, right? They are correlated. What happens with these ones? Is there any constraint about this? Like, I never know how this is in English. In Spanish would be ligadura. Or I think in Portuguese is something like that, I would say. Is there any relationship between these probabilities that makes them dependent to each other or not? Yes? So let's say if I take Q this has to be equal to 1, right? So this is going to be important when we try to get an idea of how to manage these terms. All I want you to know at this point is that, of course, the global densities, they are related to each other, and the local densities, the local probabilities as well, of course. Because, I mean, the probability that my neighbor is 1 plus the probability that is 2 plus the probability that is 0 is 1. Okay? So if, let's try to discuss the approximation number 1 that I would do to these local probabilities there. So let's say that we have this probability that I take a place occupied by 1 and the neighbor is 0. How would you approximate this probability? Do you have an idea? Like, which would be the most brutal way that you would say, I have no idea, so I would say this? So, half? Why?
If you distribute them randomly, you would get half probably that happen at a certain site at t equals to 0, let's say.
Who disagrees with that? I disagree with that. I'm going to tell you why I disagree with that. If I have 10% of zeros and 90% of ones, I don't think I have 1/2. What do I have? What do I have? You are this close, epsilon close. It's a very good intuition. 1/2, because if I'm randomly, I have 1/2. But if I don't have my densities equal, the density. Right? So 1/2 if row 0 is 1/2. Then it's correct. If not, more generally, row 0. What I'm saying here physically? What is the reasoning I have when I write this? The reason that I have, yeah, it's well mixed, but well mixed in which sense? Is that I don't care about the 1. The probability that I take a neighbor that is 0 is the number of zeros. And then it's well mixed. I'm forgetting about the spatial correlations. I'm just saying the probability that I have a 0 next to a 1 is the probability of zeros, and let's go for that. This is the well mixed. That, of course, I can write this way, no? To avoid introducing one more variable. Of course, for this term here, I have the same. I have row 0, which is this. And for row 2, 1, I have row 2. No? The density of 2s. So at the end, nice. So at the end, what I have is this is what I get if I approximate all my local probabilities Q by the well mixed approximation. This is easier, no? This is, how many of you have analyzed a system like this before? At least? Okay, this is easier, no? Now, something that is very interesting is that I have all my system described in terms of global densities. That is good, because I just have a dynamical system, two-dimensional dynamical system that I can analyze. It's bad because I don't have the space, no? So if we go back to our experiment, that is now there, small, which case do you think this approximation would recover? The liquid one, no? So in principle, I built my model starting from space, and I am taking an approximation to study one of the experimental conditions that I had. So if my model is correct, or at least partially correct, I should have, in this model, this bistability behavior that I was showing before, no? I was discussing before. So how many of you know how to find fixed points, stability of fixed points, and these kind of things? So now the class is going to be shorter. Great. So the first thing I do when I have a system of differential equations, I can try to find which are the values of the densities that stop this dynamics, which is the fixed points. And a very convenient way of doing that is by basically I can, this is row 1, this is row 2. I can define F1, row 1, row 2, that is going to be this side of the equation here. This is equal to 0. I think I'm going to use row 2 here, so you can follow the slides easy. And then from here, I can basically write row 2 as a function of row 1, and this is what I call the null clines of this system. So in this case, if you want, we can write it. Delta row 1 is going to be equal to beta 1 row 1, 1 minus row 1 minus row 2. And then this is delta minus beta 1 beta rho 1 minus rho 1 plus, I think I'm missing a parameter here. Let me see. No, I'm not missing a parameter. I'm missing something. What I'm missing? What I'm doing is something very stupid. If no one tells me what I'm doing stupid, it means that you don't know how to calculate null claims. Sorry? So one of the null claims, if I equate this to 0, one of the null claims is rho 1 equal to 0. Yeah. The other one is delta. Yeah. Who said yeah? Beta 1, 1 minus rho 1 minus rho 2 equal to 0. And then basically what I have is delta minus, now yes, delta minus beta 1 1 minus rho 1 minus rho 2 is equal to delta 1 minus beta 1, and at the end rho 2 is equal to 1 minus so basically one of my null claims is going to be rho 1 equal to 0. And the other one is going to be aligned with a slope that will be given by my parameters. I can do the same for the other equation, and I will get a null claim that is rho 2 equal to 0, and I will get another line that is given by my parameters. So what is interesting about this approach is that even though I have no idea of how my equations, how are the solutions of my equations, I can try to understand the slopes of these lines to see how they behave. And basically, whenever I have crossings between the null claims is when I have fixed points of my system, because it's when both variables are, both derivatives are equal to 0. So as homework for tomorrow, I'm going to show you all the results, but I think it's a good homework. I want you to look at the, and I'm pretty sure you all will do that. I want you to look at how, depending on the relationship between these parameters, the slopes of these two null claims change. And you will see that basically, depending on how you play with delta and gamma, and more importantly with gamma, you will see that the structure of these lines change. Okay, I'm going to show you the plots now, so don't worry. I'm not going to stop the story. Yeah, questions? Yes. What do you mean by how this is true?
You have some script version.
Yes. Yes.
And some limits a continuous variable.
So it's not a discrete and a continuous. So it's the same as going from the Hamiltonian for the IC model, where I have the product of the spins, to the equation for the magnetization.
But then you are talking about probability distribution and those distribution.
Yes.
Microscope average is connected with the microscope average. But there I feel a little bit to see.
All I'm doing is I have a lattice that has different cells in different states, and I am writing an equation to describe the macroscopic state of the lattice. To which point is true? To a very limited point. And that's all the point I want to make. For this system, I'm not going to show you the full thing, but for the contact process, we are going to see the numerical results for the density, and we are going to see the mean field, and we are going to see everything, and we are going to see how bad approximations each of them are. But yeah, it's not a good approximation. I mean, if I have spatial correlations saying that the spatial correlations don't matter, it's not a good approximation. Great. So let's focus on trying to see how these null lines behave a little bit. So when the gamma is very low, which means the killing parameter is very small, so basically I am investing a lot of energy into in producing some chemicals that do nothing to the others. In that case, here what you see with the red line is the null line for the, so colicin producer is 2, is 1, these are the producers. Sensitive is 2, are the non-producers, so these are the ones that die and these are the ones that kill. And here we have a case in which I am producing a lot of chemicals, but they do nothing to my neighbors. The red null line is the row 1 null line, and the black null line is the row 2 null line. Okay? So in this case, basically you see that when gamma is small, they don't cross. And therefore, I only have 2 possible stationary states that are the ones in which the red dashed lines intersect with the black lines. One small comment that I want to make is, so why do you think I am having a triangle only for my phase diagram or for my flow diagram? So why x goes from 0 to 1, y goes from 0 to 1, but why I'm not showing you anything here? Sorry? Because it violates my condition. So here it doesn't have any physical meaning. How many of you have seen a diagram like this before? How many of you are totally lost at this point? It's fine, it's fine. I will just try not to get you lost tomorrow. Okay, I will ask after in the coffee.
I'd say that for me, it was this plot analysis was too fast.
Okay, so basically, once I have my dynamical system, all I want to know is in which points the dynamics of 1, the dynamics of 2, or the dynamics of both is equal to 0. That means that if I define a space between my 2 variables, the values of the densities at which this will happen are going to be given by these null lines that I'm getting from here. And this is what I am drawing now here for you. Okay, forget about the arrows in the back if you want. All I'm saying is that if gamma is very small, the slopes of these lines is such that the lines don't intersect with each other in the middle of the plane, which means that I don't have any potential equilibrium there. Is that clear to everyone? The only points at which the dynamics can go in the very long term are the points at which red lines intersect with black lines. So this black line intersects with the red one here and the red one with the black one here. Why? Because those are the conditions at which rho dot equal to 0 and rho 2 dot equal to 0 are met at the same time. So these are the fixed points of the system. Is that clear? Okay. So in this case, I only have 2 possible stationary states. But now the next question is whether these stationary states are stable or not. The fact that I see, if I have a cup like this and I put a ball here, this is a stationary state. But if I kick the ball goes away because it's unstable. But if it's in a minimum of the potential, if it's in the top, it's stable. I am not going to go into how you get the stability of these fixed points, because if not, I would spend a lot of time. You can do that. You linearize the equation and you calculate how the perturbation responds. What I want to show you is that we can look at numerical simulations of the equations and we see to which point they go in my phase diagram. And in this case, they all go to the fixed point in which the producers die and the non-producers persist. Why? Because the producers are killing themselves? Because there is no space for producing. And I think the most important reason is that the efficiency is very low. So I am almost not killing anyone, and I am paying a very high cost trying to kill, and I am not killing anyone. So then I am growing more slowly. Remember, my beta 1 is smaller than beta 2. So I'm growing very, very slowly. And what's going to happen is that I grow very slow, I don't kill anyone, and therefore the sensitive ones are going to persist, no matter where I start. If you understand better this picture, I can put an initial condition, and over time what I see is that the sensitive will always grow and the producer will always die. And it doesn't matter how small they are. If I put the gray line here, it grows up. I can send you the mathematical notebook if you want to play around with this. It always grows. And I already see we are facing because this is not the experimental result that we saw. Yeah, but we're in a limit, no? The limit of a small efficiency. So don't get mad yet. Let me show you this figure first. I think that's going to then help understanding the phase diagram. Now I have the high efficiency case. Same initial conditions as before. Here I have something wrong with the, it's the same figure as before, no? Eesh. Okay, let's go quickly through this. So you see, this is why you shouldn't, well, I will show you tomorrow the trajectories. What is important now, I want to, the point that I want to make is that now if the efficiency is high, I tune this parameter gamma in my model here. Now you see that the null lines, the slopes are changing, and I introduce a new fixed point. There is a new point in which the two dynamics freeze. And now, depending on the initial condition that I set for my model, the trajectories will go to one side or will go to the other side of this fixed point. This one is a saddle node in 2 dimensions. So the key message here is that I have one control parameter in my model that is the killing, is the efficiency of the killing. And playing with that parameter, when the killing efficiency is high, I can explain my experimental result. I have this bistable behavior when this killing behavior is highly efficient among these two types of strains. Okay? Remember, this is all well mixed. So questions? Yes?
The parameter is efficiency because it increases interaction.
It's because when gamma increases, the depth of the row 2 is higher. So it's some sort of aggressiveness or killing efficiency if you want to think in those terms. More questions?
So the low efficiency is like the plot for the plate experiment.
Yeah, but which determines the behavior? The low efficiency, what it's telling me is that the non-producers always win. It's explaining, is not explaining any experimental result, right? Why?
Producer should win, right?
In the lattice, yes. But why do you think that the low efficiency is not explaining any experimental result? One by one. Because evolution will not allow any behavior in which you invest in killing and doesn't kill, basically. Right? So this is the second part of analyzing these kind of models. The models can give many things, but of course, you are bounded to the parameter regimes that make sense, no? A very reasonable question and a very interesting one is which are the parameters that make sense, no? Here I'm being extremely qualitative. I'm saying high efficiency, low efficiency. You can do that, like you can try to understand the general behaviors of the model depending on the regimes, but you can also do experiments and parameterize your models, no? More questions?
This case didn't exist, but
No, this does. This is the experiment, no?
sorry, the low efficiency case, but or it exists, but it deaths immediately.
That's a very good question. That's a very good question that maybe is not for the scope of this course, but to which extent it's, but what do you mean by it exists but it dies immediately? Who dies immediately?
The case of opposite of evolution.
You can have any kind of mutation that favors this kind of trait, this kind of behavior, but then it doesn't fixate in the population, if that's what you are asking. If it can appear or not, in principle, yes, you can sample the space of possible behaviors, but only a few of them are successful, no? And of course, having a trait that makes you reproduce more slowly and doesn't give you anything in return will not appear in a population, no? So there is no reason for having a killing trait that doesn't kill. But in principle, it can appear and disappear very fast, no? It's eliminated. If there is any biologist in the room, take with care what I'm saying by itself. Well, because it can be fixed randomly and yeah.
Is efficiency given only by gamma? For example, if beta 1 came closer to beta 2.
Yes, great question. The high gamma limit, or let me rephrase that way, the small gamma limit and the beta 1 almost equal to beta 2 limit are identical. If you write the equations, they are identical. So in these small courses, I usually don't like opening all the math because I don't like spending 1 hour doing algebra. If it's something that you really need to understand what I'm saying, please tell me. I told you I wanted this to be informal, and I want to be informal in the sense that I want you to learn. If you think that it's important for you to learn that I open the math in parts that I maybe don't think is important, tell me, and I open it. I have no problem. Okay, but I don't want to spend 20 minutes simplifying equations. And it's totally fine. I don't expect that today in the afternoon you go to the hotel and you open the math. I expect that today after the project you go and have some drinks with all of you and you make friends and you, but when you are home, if you are interested next week, you can open the math and you have any question, you can write me and I can tell you what is the thing. It's totally fine. But I don't like opening the math because, yeah.
Maybe it's interesting for you to show the result just so we can know which parameters define high efficiency.
Yeah, well, high efficiency, okay. Let me write the null lines. Maybe if I write the null lines all the way, it's easy. So if you set your two differential equations to 0 and you solve, so you write rho 2 as a function of rho 1, this is what you get. You get one nullcline that is basically extinction of one of the species, the two axes here, and the other nullcline are these lines that have always negative slope, and when rho 1 is equal to 0, they have some crossing value with this axis here. Okay? When you manipulate your experimental parameters, beta 2, beta 1, and gamma, all you are doing is you are playing with the crossing points of these nullclines with the axis because you're manipulating the slope. Right? So for instance, what happens with, let's say I take this nullcline here. When rho 1 is equal to 0, this is going to give me this crossing point here, right? If beta 2 is higher, this goes to 0, and this crossing point goes to 1. When beta 2 gets smaller, this is going to start going down, 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 down. Of course, it always has to be positive. It cannot be negative. So basically, when you play with your growth rates and your gamma parameter, and you can also play with delta, what you will see is that you are stretching these crossing points here in a way that these two lines are going to be either this or they're going to cross. So you can fix all your parameters and just play with gamma and see what happens with these lines when gamma goes from 0 to very, very high. Helped? It helped a little bit? Yeah. Okay.
So there is never a range of parameters in which the species coexist, right?
Here, no.
Okay.
Here, no. So let me, just to finish, making the point of what are the parameters that control this behavior. You can do all this analysis in a more extensive way, and basically you can see that depending on your parameters, you will have a region in which you have this bistable behavior and a region in which you lose your bistable behavior. And you can even know which is the threshold that we were defining in the beginning for producers to go up or go down. Okay? So once you know the behavior, you can, of course, analyze your parameter space more broadly. And this is what they show in this paper. They show that basically when the cost is very high or the effectiveness or efficiency is very low, my system, I don't know why I do this, sorry. My system is always stable, monostable, which is the limit that we were saying before. But when the cost becomes smaller, smaller, and smaller, or the effectiveness increases, then I enter into the bistability region. And if you take one combination of these two parameters, then basically you can define what is this critical frequency of the producers that makes them grow or go extinct. Right? So again, this is all for well-mixed models. This is all differential equations. I am assuming that the spatial correlations don't play any role. That's why it's completely different to my lattice. Okay? Good. So what we're going to do tomorrow, I'm going to show you a little bit the, actually let me use the 10 minutes that I have to finish this story, and tomorrow we go into them into a little bit more of the matter. Once I know